Okay, let's continue. Now we are going to see another variation to pairwise sequence alignment. By the way, the first version, the global sequence alignment, the dynamic programming solution was first proposed by Needleman and Wunsch. So that's the algorithm. When you hear Needleman-Wunsch algorithm, it's the dynamic programming solution to the global pairwise sequence alignment. So it's called the Needleman-Wunsch algorithm. Now we are going to talk about a modification for the local sequence alignment problem, which was proposed by Smith and Waterman. So it's called the Smith-Waterman alignment algorithm. Is the same dynamic programming solution, but this time for local sequence alignment. So why do we need local sequence alignment? This slide describes that. If you want to align, we may have both of the sequences long. For example, we may have a 4,000 base pair, and it's compared considerably shorter than the other sequence. We have a long DNA sequence, and we want to compare it with the complete yeast genome, which is 12.5 megabase length. So what if only a portion of our query, say out of this 400 base pairs in our gene, maybe there's a domain which is exactly shared, conserved in the yeast genome. Okay, so imagine there's a 200 base pair exact match, or or it's very similar, a strong similarity to a gene in yeast. So either using the global alignment or semi-global alignment, we would not find this subsequence that is a match, because while doing the alignment, even in semi-global alignment, it tries to match the complete sequence, okay, the complete 4,000 base pair sequence, and when it's computing the score, it's going to have lots of mismatches. So these mismatches, intermediate, so look at this, it's like only 5% of the sequence is matching. So this 4,000 base pair, this 200 base pair is like 5% we are interested in this 5% region, and this 5% region is matching, but 95% of the sequences do not contain a match. They are just random sequences. And in that case, we will not be able to find it because of these 95% of the sequence will have lots of mismatch penalties, and we will not be able to find this matching region. Because again, our goal is to try to globally maximize the score, so we may oversee this match. Now, what we need is an algorithm which will find us these sub-regions in both of these sequences. If there's a subsequence in both of these sequences, if there's a subsequence that is matching very well, can we find this subsequence? So that's the local sequence alignment problem. Not interested in the whole sequences. Can we ignore parts of the sequences and find only a sub-region, subsequence in both sequences that are matching with each other? Okay, the problem is defined like this. So now, so here's an example, X and Y. So so this is the formal definition. Given two strings of length capital M and capital N, find substrings X prime and Y prime whose similarity, in other words, we are globally, optimal global alignment value, we are going to globally align these substrings, okay? That's the difference from the original global alignment problem. The global alignment tries to globally align the whole strings, okay? All of them are matched up to 1 to M and 1 to N. Now we are interested in substrings X prime and Y prime in these sequences, and we are trying to find such substrings whose alignment score is maximum. How do we find such subsequences, and how do we find this score? For example, look at this CCCGGG is an exact match, so we need to find the longest sequence, it's not the longest common subsequence problem, because the longest common subsequence problem in computer science, or longest common substring problem, is interested in having an exact match. Okay, I mean this example clearly shows an exact match, but sometimes we may have mismatches. This G may be A here, but still this sequence, these subsequences may have the highest optimum score. Okay, so we what is the subsequence in both strings x and y, which will give us the maximum score? Okay, we cannot say shorter sequences. The shorter the sequences are, the less matches they are going to have, right? So we try to increase the length of the sequence. If there's a match, the score is also going to increase. But the mismatches, when the length is increasing, we may have mismatch. Our mismatch probability may increase as well. So the score is going to decrease. So we want to find the place where the score is maximized, and only report that subsequence which receives this maximum score as the alignment. So now this modification is going to be less trivial compared to the semi-global alignment. So how can you, this is another picture which shows what we are interested in. So there may be a certain path which ends at score of -10. And this subpath here, the green path, may have score 50. And this last part may have score of -5. Now we want to report an alignment score 50. Okay. And saying that, match these green subsequences. If you match these green subsequences, you'll get a score of 50. Okay? But think about this. If we were to, so how are we going to modify this dynamic programming recurrence relation? I mean, if we were to add these things, even if we come here, the original dynamic programming would give us 40, right? Because this 40, what is the score? The score of aligning the prefixes. So it contains the blue regions as well. So we may have, if we stopped our alignment here, you may say that, okay, ignore the rest which gives you the negative score, ignore that part. But we also need the mechanism to ignore the first part as well. Because there's a subsequence which is in the middle, which is a score of 50, but this is like, this does not agree with our definition of this f, okay? F was the score of Fij, and each cell entry, Fij, shows us the best way of aligning the first I characters with the first J characters, so it also includes the blue part. So, I mean, the entry here at this point, Fij, would be 40, okay? But how can we get rid of this -10 region and say that we can actually have a score of 50 here if we started our alignment from here, right? So this is a hint for you. We need a change in the dynamic recurrence relation which will allow us to start an alignment fresh at an intermediate point. So what modification would we have on the Needleman-Wunsch algorithm? So what did Smith and Waterman propose? Any guesses? So it seems like a very difficult problem, but the solution is extremely simple. So let's get back to the original Needleman-Wunsch recurrence relation. So what would you do? How would you modify this one to solve the local alignment problem? Okay, let me tell you. If yeah. But which position? I mean, you're saying is subtracting is minus 10, but it will, if you subtract minus 10, it will give you 60. I mean, at the final, from 40, if you subtract it, it's going to give you 50, right? So how would you find out which position to subtract? Because at that point, when you come to that 40, at that point, there are many previous cells that you can consider. How would you find which cell to subtract from? But that's a good, that may work. I mean, maybe you may trace back up to a certain point, and during this trace back, you may try to subtract one of these. Yeah, it may work. But the actual solution is much simpler than this. You don't need to do any trace backs, nothing. The solution is just in this maximum, look at this maximum equation. Just in addition to these three options, just add an option of 0. Okay? If you just, while looking at the maximum, just add a fourth member, 0. What is the meaning of that? It means that if the alignment score falls below 0, you can opt out. You can just say, I'm not going to choose this alignment. I'm going to choose 0. Start an alignment fresh here. I'm not going to allow negative. If my alignment score falls negative, it means that I'm in a, this leads. Why not start a new alignment from there? If this alignment up to here has a negative score, then just ignore that part. Start a new alignment from 0. This solves the problem actually. It solves optimally. Just having the option. So in your table, you are not going to have any negative values. You will have zeros, and the 0 at an alignment entry is going to mean that you can start a fresh alignment starting from that ith and jth character. Okay, so this simple addition by just replacing a certain negative number here, when you reach the certain negative number, you can put a 0 at that point. It's not the same as, this modification is not the same as replacing all negative numbers with zeros here. Okay? You'll get a different table then. For example, the thing is, what I'm trying to say is, replacing this with 0 will maybe make these numbers, the cells next to it, positive. Okay? So it's not like looking at a completely processed Needleman-Wunsch table and replacing all the negatives with zeros. You may end up with a 0 score at the end. But in local alignment, if making this 0, for example, making this minus 15 0, maybe you can have positive values. For example, this will be 3. If this was 0, if this minus 15 was 0, if we could start a fresh alignment at this point, starting from this H and starting from this, I think W and W, or sorry, H and W, we could have a score of 3 instead of this minus 12 here. We could have a score of 3 if this place was actually. I think this is coming from a gap, but let's look at this one here. Minus 19 to minus 4. I mean, the score of aligning W with a W is minus 15. It's a plus 15. Okay, so it's a plus 15 from minus 9. So if this was 0, we could have a plus 15 score here. We could have started a fresh alignment with at W's. We are going to have a plus 15 score right at the end of aligning with W with W. So at the end, we would have a higher chance of getting large scores. Now, but is this modification enough? Having 0. What about the final optimal score? Is it going to be at the bottom rightmost entry? So what is the optimal score of the alignment? We have filled in the table, we have completed the table by having this modification, inserting zeros instead of negative numbers. We have the choice of 0. When the alignment goes below a 0, we have the option to insert a 0 there. And what about after we complete filling the table, what is our optimal score of local alignment score? It's the maximum number in the table. Okay, it's not necessarily, the maximum number in the table is not necessarily the right bottommost entry. It could be anywhere in the table. So this modification of adding a 0 to this maximum allowed us to start. So I double quote, emphasize the word starting. We could start an alignment anywhere. But for local alignment, we should also have the option to end the alignment anywhere. Which means that we don't need to go all the way to the bottom rightmost entry. So we could have stopped an alignment, so the number, the matches increase, and if at a point we start getting mismatches, the number may decrease still above 0, but when it starts to decrease, we may say that, okay, I stopped my alignment where it was maximum. I don't have the mismatches at the other end. So in order to have the freedom to stop, to end an alignment anywhere, what we do is, the modification we do is, we just look at the final table and look at the, so this is what we do. This is the only modification is addition of the 0 to the recurrence relation, and the initialization of the first rows and first column are obviously, since we do not have negative numbers, even if we have a gap penalty, gap penalties are negative, so we choose the zeros there. So this is how we initialize the first row and first column. And here is a filled-in table. Okay. After we look at this table, we look for the maximum entry in this table, which is 14 here. Okay. So it says that my local alignment ends after aligning this F with this F. So I ignore the last two amino acids of both sequences, GY and KL. Even if I have a positive score at the end, okay, 10. 10 is a positive score, but there was a better score, 14, if you ignored the last two mismatches. Okay, so here, this example does not use the Blossom matrix. It just says matches 4, mismatches -2. Okay, and here's how we fill in the table. We start with zeros, and if you look at this K and E, if you take the gap, you are going to still stay at 0. If you take the mismatch, it's still 0. So 0 is the maximum out of these negative numbers. So as you can see, when you're filling in this table, if any option drops below 0, you just put a 0 there. Okay, 0 is our fourth option. Now you may think about what happens when the traceback. Okay, this 0. We did not come to this 0 either from none of these, okay? When we are doing traceback, what are we going to do at this point? We're just going to stop. This is where our alignment ends, right? And you don't need to do a traceback from 0. This is where, when you reach a 0, this is where your alignment stops. This is where your alignment begins, actually, when you reach 0. So what we are going to do is, we are going to start our traceback from 14 and say that, okay, where could that be? So let's do the alignment here. Okay, this is, if somebody gives us this table, your job is to write down the local alignment. What is the local alignment if you're given a table like this? The first thing you do would be to find the maximum number, which is 14, and then from 14 we need to do a traceback. Okay, now to find the last alignment position, we need to answer this. How did we obtain this 14? Did we obtain from left or top cells, or coming from the diagonal? I mean, left and top cells are 9. They are numbers smaller than 14. And we have a gap penalty, so gaps cannot increase this number 9. So we came from 10 to 14. Since we have these F's at the end, F is a match score is 4, so it is consistent with the table. So a match score of aligning these F's at the end increase this previous score of 10 to this is a match score of 4. The overall score of this local alignment is 14. Okay, this is what I do not right now when I'm doing traceback. I'm at this point. I do not know right now how this 10 is obtained. Okay, but I'm going to find it out by going one by one. But this is how the score, what the traceback tells me. So this is the last position, and after 10, again we are going to the diagonal from 10 to 6. Okay, what we have is E's. We have a match of E. E is matched with an E, and the score before that was 6. So 6 plus 4 gives me 10, and then before that I have L's. L's are again matches. So, and right now I'm here. I have gone diagonal from here to here, here to here, and here to here. Now, what I have here is a 2, and I have 3's here and 4 here. And if you look at this 2 could have been obtained in any of these 3 cells. Because gap penalty is -1, we could came from top, from left, or from the diagonal. Because the mismatch penalty is -2. So this 2 is consistent with coming from all these options, you get 2. So it means that our alignment actually can go 3 ways at this point. So it was V and A. If we choose the mismatch path, so sorry, A was the top sequence, so A and V. If we take the mismatch path, we have a mismatch like this. So we are at 4 right there, and then from 4 to 0, we have a KK match. Okay. So this is the optimal, one of the optimal alignments, which results in a score of 14. And you can verify that. 4 matches, 4 times 4, 16, and 1 mismatch, the final score of 14. So it is consistent with your score, and this is your local alignment. As you can see, we have ignored the first 4 characters of the first sequence and the last 2 characters of both of the sequences. So this is the local subsequence which gives us the maximum score. And the other 2 options, let's look at the other 2 alignments. If we have taken the top path, in the top path for example, the top path means that, going to the top means that from V to A, we are still at A, so V is aligned with a gap. And after we are here at the top, now we are, if from here, if you choose the top path, means that you first align V with a gap, and from here 3, you could not have come from this way or this way, okay? We do not have a match score of 3, matches 4, so we do not choose this way, we mismatches. So the only way from this 3, the only way to go is to the left. Okay, that's the only possible way, which corresponds to aligning a gap against an A. Okay, then from that 4, again, we are in the same spot, we go to this K. So the other 2 options, instead of these, since mismatch score equals 2 gap scores, it means that this region has equal penalty if I do this, if I do that, or if I do this. Okay, both of them, and this one corresponds to going left first, then top second. Okay, these are the three local alignments which give, which all of which are optimal. Okay, so it shows one of them. You could have chosen these show the other two. There's another example. I leave you. I leave this as an exercise for you. Okay, here these two DNA sequences which are of similar length, and we have a match score of 4, mismatch score of -4, and a gap penalty of -4. So gap penalty and mismatch scores are the same here. And actually, here's the quick. The solution is 22, which is this. You may try to fill it on your own as an exercise. It will make you, if you practice, you'll understand the algorithm better. And here's some actual results from real sequences that compares local with global alignment. And this is the similarity score here. The global alignment we have, it compares all three options, okay? And this is the hemoglobin beta chain human versus the other sequences. So we have the hemoglobin beta chain. If you compare the sequence by itself, you get all matches, okay? A self-match, so it shows this score for reference, okay? It's like the maximum score you get was 725. If you had all matches to this input sequence, this is the maximum score you can get. And whether you hit local, semi-global, or global, and it's the same alignments you get. So you get these same scores. If you compare it to the hemoglobin alpha chain, okay, both of them are hemoglobin proteins. What do you call these two proteins? Alpha chain and beta chain? What was the term for? Not orthologs. They are both human proteins. Yeah, they're paralogs. These two proteins are paralog proteins. As you can see, they are paralogs, but there is a lot of divergence in their sequences. They are not very similar. It's not like there are a lot of mismatches, mismatch in amino acid, because it shows that the alignment score is 314 compared to the maximum we could get, 700. It's like 50% sequence identity or so. So it's, we lose more than half of the scores. And in that case, since these sequences are related, alpha chain, and in this 50% similarity, they are of similar length. Most of the sequences are matching. So whether we do global alignment, semi-global alignment, or local alignment, we do not lose much. Okay, they are similar length, similar sequences. So we do not need local alignment here. Actually, we could do global alignment, and the scores are similar. However, if you look at this, the chicken lysozyme precursor. This is another protein which is found in chicken, and the global alignment gives us -107. Okay, it's a very bad score, lots of mismatches. And no end penalty, we get a positive score, but there's a local match which receives a score of 32. So there's a local region in these hemoglobin beta chain, and the chicken lysozyme precursor, which actually has a small motif matching in those sequences. And more dramatic ones are the calmodulin protein in human compared with this calpain heavy chain. Again, it's a human protein. They do not match at all. I mean, it's like -2000 as our score. But if we do either semi-global alignment and local alignment, actually, probably one of these is very small protein compared to the other one. Actually, it can find a better score compared to these local global alignment. Okay, so these shows real examples, and most of the cases we do not. These examples do not. It's not like, since they are both proteins, we do not have one of them very long, the other very short. They're of always comparable sizes. The benefit of local alignment with respect to semi-global alignment would be more apparent if we were comparing a gene with a genome, for example, the complete genome. In that case, even this may be, or multiple genes, a certain genomic region against the whole genome. If we had done that search, the semi-global alignment would get much worse scores than local alignment. But in this table, it seems they receive comparable scores. Even this case, this 500 is not very bad compared to this 655. Now, since, I mean, it's a polynomial time algorithm. Polynomial time algorithms are considered efficient algorithms, okay? If you have a polynomial solution to a problem in computer science, it's considered efficient. It's doable, okay? It's tractable. Here, for these dynamic programming solutions the table requires m times n entries. So we need mn space. The space complexity is ONM, and the time complexity is also OMN, because to fill in each table entry, we need constant number of operations. What we are doing is looking at 3 choices. This is always constant. For each table entry, we have O1 operation. And there is a modification to this dynamic programming solution in terms of space. If we only need the score, okay, if the alignment itself is not needed, you may not need the alignment itself. For example, if your goal is to find the most similar protein in a database of proteins, you're not interested in the alignment. You may want to compare your protein to all of the other proteins in the database and just retrieve the one with the highest score. You're just interested in the in the protein itself. You may not be interested in the actual alignment, and in that case, you don't need to use the whole table. You can, if only this maximum score value, not distance, or can be replaced with maximum score. If you're only interested in the maximum score, there's a modification to this algorithm which do not use all the table entries. So at each row, there's a linear space solution, not linear time. We still need OMN time, but it's a linear space version. We can discard some of the table entries if we are not going to do a traceback. And there are more complicated divide and conquer algorithm that doubles time complexity, but it's still quadratic, okay, and uses much less space, minimum of these 2 sequences. But as you can see, these problems are studied very, very long ago. I mean, these sequence alignment, the first bioinformatics solutions, it appeared more than 30 years ago. Okay, so this is considered efficient in general. In computer science, we have problems which require exponential time or space complexity. So the polynomial we should feel like, and especially O(n) square, shouldn't bother us that much, or so. Or should we bother by this? I mean, should we just say that, okay, this problem is solved? We don't need more efficient algorithms, or this cannot be made more efficient because maybe this may be the lower bound in terms of complexity. If you want to find 2 sequences, you need to consider at least a pair of them. So m times n is the smallest you can get. You cannot get better than this if you're looking for optimal solution. But if we are stuck with this OMN time, here's an example which shows you how much memory and how many days you're going to need if you're comparing 2 small size genomes, okay, not large size. These 2 1 megabase genome means that 1 million nucleotides, 1 million base pairs. And do not forget the humans we have 3.2 billion letters. Okay, 3,000 times larger than one of these sequences. So this is 1 million. So human genome is 3,200 times larger than one of these sequences we are comparing. Okay, so if we have a bacterial genome, maybe 2 bacterial genomes, each of them are 1 million character long. If we use Needleman-Wunsch global alignment or Smith-Waterman local alignment to align these 2 genomes to find out whether these 2 bacteria are coming from the same strain, or I mean they are a variation of a certain, maybe a virus, maybe a viral genome, you may compare whether they're related to each other. And here, if you use an integer to store your score, okay, to store your score, you may use an integer. An integer would require 4 bytes, okay, if you implement it in C, for example. In GNU-C, the integer is 4 bytes. Even if it's 1 byte, okay? Even if you imagine your score does not exceed 255, you use an unsigned character, which is 1 byte, you can represent up to 255. You would need 1 times 1 million times 1 million, 1 terabyte memory. Okay, 1 terabyte of memory, it's still, I mean, even if memory is getting cheaper, CPUs are getting faster, but it's still 1 terabyte of memory, it's still large, right? We have like 16 gigabytes or 32 gigabytes, 64 gigabytes of memory maybe there, but 1 terabyte of memory is still out of reach in even today's standards. So you would not be able to store your table in the memory. You need to write it in a file. You need to read your scores from the file. So you need to write your scores in a file. And that file you can store in your hard drive, but it was 1 terabyte. You need again, even the hard drive, you need a large hard drive. I mean, 1 terabyte is a lot of space. And in terms of time, if we use a 1 gigahertz CPU, I mean, imagine you have 4 gigahertz CPU, it would take 2.5 days. Okay, so to fill in this table, if we have this many entries in the table, 10 to the 12th entries, it's going to give us, so it's going to take 1 million seconds, which is going to take 10 days if you're using the 1 gigahertz CPU. Yes?
Isn't the time of input outputs memory to hard drive?
Yeah, this is the optimistic case, and reading the sequences, writing, and it may increase. These are just optimistic guesses. You would need at least this much. Okay, so I mean, just imagine we have many bacteria. We want to find out the relationships between 100 bacteria. Pairwise alignment of these 100 bacteria, with considering all pairs of them, it's 10,000 pairs in 100. If you have 100 bacteria, it's going to make about 5,000 pairs. Okay, 100 choose 2. So it's going to, you need maybe a year to make such an analysis. So even this efficient considered algorithm is still cannot be useful to us if we are going to compare things other than just proteins. If you're comparing things like 1,000 characters, 1,000 sequences, you can use these. But if you're going to compare large whole genome alignment, this dynamic programming sequence alignment is going to take a lot of space and a lot of time. Another thing is, even if you're not doing whole genome alignment, imagine this. You are doing a database search. Okay, this is the most common things biologists do when they discover a new sequence, is to search the new sequence against known sequences. Imagine we have found a sequence of length 400, and we are going to align it against, for example, 5 million sequences. In our database, we have 5 million protein sequences. There exist 5 million sequences. And each of them, on the average, again, also is length 400. Okay? So, this search, let's try to find out how much. So what we do is, we compare our query protein to each of these 5 million proteins. So each alignment is going to take 1 square time, so 400 times 400. So it's like, so this many operations, so it's constant. I'm not going to use O notation. So I'm going to need this much time and space. So 16. So 160,000 operations for one alignment. This is not much. I mean, it can complete in seconds, maybe much less than a second, because we can do 1 million operations in a second. So it's going to take like 1/10 of a second. Okay, but we are not. This is for one comparison. We are going to do 5 million comparisons. Okay, this is for one protein in the database. We are going to do it for 5 million times. So 5 million times this. So we could do 1 million per second, right? We could do 1 million operation per second. So it's going to take this much seconds. 5 times, so it's going to be like 800,000 seconds, right? 800,000. No, it should be much less than. Actually, it's very close to. No, it's not. It shouldn't. It's not. It may not be less than that. And this 800,000 seconds come very close to our whole genome alignment problem. So it shows you that even if you're not doing a whole genome alignment, instead of comparing 2 genomes, even if you're doing a database search of a small sequence against millions of small sequences, you're still stuck with your running time. And maybe you're not going to need that much space, because after you do one alignment, you can throw away the tables. Okay, you're not going to need that much space, but for sure, you're going to need a lot of time to do a database search like this. So you don't want to wait 10 days after just issuing a query. You get your result by email after 10 days. This is going to, and for one query, this is unacceptable. So what people have come up with is for such database searches, they've come up with heuristics. Heuristic means that algorithms based on some intuitive ideas, but they do not guarantee optimality. Okay, that's the main thing. They are very fast, but there's a chance that the results you get may not be the best solution. You may oversee the best solution in the search, or you may not exactly find what you're looking for, but most of the time it works. Okay, so for example, for 95% of the sequences, you would be okay. If only 5% of them you miss such kind of a match, you would be okay with that. Because this is optimality versus speed. So if instead of waiting 10 days, if you get reasonable results within seconds, you would prefer that, right? I mean, if that algorithm gives you completely garbage results in 1 second, you would not want that. So it should give something meaningful, and that's what BLAST and FASTA does, and that's what makes them popular. Okay, that's similar to Google. They can do. I mean, their speed actually increased in the last 5 years. They are probably using the parallel cluster or cloud computing kind of architectures. So their speed actually increased a lot. The BLAST searches, it would take a minute, like maybe 5 years ago, it would take a minute to complete the search. Now you can get your search results within seconds. Okay, their speed actually, even the database size is increasing every day. Still, they can give you faster searches. So the BLAST is the short, it's acronym for Basic Local Alignment Search Tool. As the name implies, we're interested in local alignments in BLAST. It finds local similarities between sequences, and it was developed the original BLAST was in 1990, and there was BLAST 2 and SciBLAST. There were extensions to this BLAST. BLAST for nucleotides, BLAST for proteins. MegaBLAST is another version of BLAST which does whole genome, which is capable of doing searches for very long sequences, very fast, but with some reduced accuracy. Okay, and it's basically designed for, specifically designed for database searches. And the main idea is that a good alignment between two proteins contain short lengths of exact matches. So it's based on finding exact matches, which is called a seed match. So finding an exact match can be done very efficiently with the use of hashing techniques. Okay, because we could not use in dynamic programming. We were looking for, we could have mismatches anywhere within the sequence. We are not looking for exact matches in the dynamic programming solution. We may have mismatches, we may have gaps in between, but the BLAST says that if I can find an exact match, exact substring, which matches exactly in these two sequences, if I can find this very fast, I can compute an alignment by extending these seed match from both sides up to the scores drop to a certain threshold, and this is what BLAST does. Okay, it finds a fixed size, which is called the word size or something like that. So it doesn't, while it's looking for these fixed matches, it doesn't do for arbitrary length. It basically indexes all the database entries by sliding a window over the sequences, by sliding a window of fixed size. For DNAs, I think the window size is 11. Okay, so let's see that. Yeah, 11 for DNA and 3 for proteins. So it slides a window of size 11 from left to right, overlapping windows, and it indexes these keywords in a database. So what we can do is, these are exact matches. If our query contains the exact same 11 length word, we can say that, okay, we have a match to this protein. We have a seed match of size 11. So why is this a heuristic? Because if this assumption does not hold, okay, it's based on this assumption. If you may have a very good alignment in which we may have a mismatch in every 5 letters. Okay, if you have a very good alignment, but you have a mismatch in every 5 nucleotides, BLAST will not be able to find this. Okay, because BLAST is looking for exact matches. If you have a mismatch every 5 nucleotides, BLAST will not be able to find your matching sequences. This is how it misses certain matches, because it's based on this idea. We need to have an exact match of length 11 at least to start our alignment. If your pairwise sequences do not contain this, BLAST is going to overlook such alignment. And the idea is basically this. The slide a window and finding these exact matches. These are called seed matches, and it extends these seed matches in both sides until the score drops below a certain threshold and reports. And then the other most important property of BLAST is that it computes, in addition to the score of the alignment, it gives the user a statistical significance score, which means that since we are looking for local alignments, a biologist naturally asks this question, okay, BLAST reports me this alignment, but could this local match be obtained by chance alone? Okay, so is it something really biological similarity going on here, or is this alignment, because you have 5 million sequences, a lot of them, okay? Out of these 5 millions, I mean, even if some matches, maybe like winning a lottery, you may get a match, okay, just by chance. So BLAST gives the user also this feedback. What is the probability of obtaining the same match by chance. Actually, it gives the number. It says what BLAST gives is the e-value. We are going to talk about that next week. E-value says that out of these 5 million sequences, how many of them could have given you the same score, same or better score by chance. For example, an e-value of 3 means that, okay, I've given you this match between these two proteins, but there are 3 other proteins in the database which could have obtained the same score by chance. Okay, now, it doesn't mean that there may be, of course, better proteins with, but this is very important. With no biological relatedness, 3 proteins could have gotten the same score. So an e-value of 3 is not a very good thing. So we need people use an e-value of 10 to the minus 3 usually as a measure, as a cutoff value to indicate real. So it's 1 in 1,000. Means that there was a 1 in 1,000 chance, 1 in 1,000 number of proteins. So e-value is a number, but it can be floating point numbers. So it can get smaller to like 10 to the minus 10, 10 to the minus 20, or even 10 to the minus 300, or it can be 0. If e-value is 0 means that it's very significant. There's 0, I mean, it's what the match you get is a complete biological relevant protein. Okay, so let's stop here, and we are going to finish BLAST and FASTA quickly. I'm not going to go into the real details of BLAST and FASTA. If you want, you may read the papers of BLAST and FASTA, but I'm just going to give an overview next week in the first hour, and in the second hour we are going to start the statistical significance. How do they compute this e-value? How can we compute the chance, the probability that to obtain matches in a database? Okay, see you next week.